Hello everyone, my name is Abhishek and today I will be explaining one of the most widely used techniques in nanotechnology and particle characterization, which is DLS, dynamic light scattering. This technique plays a major role in analyzing nanoparticles, proteins, polymers, emulsions and even many products which we use in our daily life. In this presentation, I will be first covering what a DLS is, how it works, its working principle, various components of the DLS, its application, advantages, disadvantages, and at last we will also see its relevance in our daily life. So first let's see what a DLS is. So DLS is a technique which is used to measure the particle size. Particle size typically in the range of nanometers. They range from 1 nanometer to few microns. Like when particles are suspended in a liquid, they are constantly moving. This movement occurs due to the Brownian motion, which is the random motion of particles caused by collision with molecules of the surrounding liquid. DLS detects these fluctuations in light intensity caused by the Brownian motion, and these fluctuations are used to calculate or to know the average particle size in the given sample. And there are various names by which DLS is known as, like PCS, photon correlation spectroscopy, and second one is QELS, quasi-elastic light scattering. This whole technique is used for studying of nanoparticles, proteins, polymers, emulsions and colloidal suspensions. The main principle behind working of DLS is entirely based on Brownian motion and the scattering of light. Like particles suspended in any liquid, they move randomly due to the collision of these particles with the surrounding fluid, and this motion we all know is known as Brownian motion. And when a laser beam hits these particles, these moving particles scatter the light in all directions. Since these particles are continuously moving, so this scattered light intensity changes continuously, and these intensity fluctuations are recorded by our photodiode or PTM. And as we all know, smaller particles, they move faster, so they cause very fast or rapid fluctuations, and the larger particles, they move very slow, they cause slow fluctuations. And to extract the size information, a mathematical process called autocorrelation is used. Autocorrelation compares the light intensity at different time intervals. The result of this analysis gives us the hydrodynamic diameter of the particle. Parts of DLS equipment. First is a laser source. It provides monochromatic stable light. Then we have a sample cell in which the nanosamples are held. Then next, optic or beam splitter. It directs laser into a sample. Then we have photodiode or PMT. It captures the scattered light at some fixed angles. Then we have correlator. It processes fluctuations to calculate particle size. And at last we have computer placed, which has certain softwares. It displays particle size and also PDI. And we can see in this diagram we have this laser source. Then this laser source is directed into the cuvette which contains our nanosamples, and we have our photodiode or PMT at certain angle, and this correlator is used which captures the fluctuations at different times to get the particle size. And here we get intensity versus time. The light blue curve represents particles having large size, and the dark blue curve represents the particles which are smaller size. And with the help of our correlator, we can plot various curves, as we can also see here. Next is working principle. So let's see how this DLS works. So first we have a cuvette in which a sample containing nanoparticles is placed. Then a laser source which is used to pass the laser from this sample containing cuvette. Then a moving particles scatter light at various intensities. Since the suspended particles, the sample contains both type of sizes, like very small size and very large size, and since these particles are continuously moving, so the fluctuations also vary. We all know smaller particles move faster, so the rate of change of fluctuation for case of smaller particles will be high, and for case of larger particles they move very slowly, so rate of fluctuations will be lower for case of large particles. And these are our correlator or PMT. It measures the time wave fluctuations, and correlator analyzes scattering patterns. And the software which is used in this computer, it is used to determine the average diameter of particles in that given sample, and it is also used to get size distribution graph, and it is also used to get PDI. PDI is a parameter which shows how well the size of the particles is distributed in the sample. If a lower value of PDI, it means the size of particles in the sample is almost same, and if a higher value of PDI, it means the particle size varies in the sample. Next, we have various advantages of DLS. Like, it is very fast and it is non-destructive. Non-destructive means it will not harm our sample. There are various methods by using which our sample get harmed, but DLS is non-destructive, and it is very accurate. It measures almost 1 nanometer to few micron particles, so it is very precious and accurate, and it also requires very small sample value. It is highly sensitive, and we can repeat the tests. It is very sensitive and repeatable. It provides valuable parameters like average size diameter in terms of hydraulic diameter. It gives us PDI and size distribution curve. It is very easy and simple to use, and sample preparation is also very easy. And there are some disadvantages or challenges using DLS. First, it assumes that all particles have the same spherical size, and these DLS is very sensitive to dust, impurities, air bubbles. So sample need to be very pure. Then next disadvantage is it has a very poor accuracy for highly polydispersed samples. So if our sample contains particles of varying size, small, medium, very large, so it is not that efficient in calculating the size of the sample. And next one is multiple scattering can distort results. If we were using the DLS again and again, then it can distort our results. We will not get the accurate particle size. And the major drawback is it gives only size of the particle. It cannot identify the chemical. There are various applications as listed here also. Water treatment, in food industry, studying of emulsions, pharmaceuticals, nanoparticle drug delivery, protein aggregation and stability, chemical or polymer labs, polymer size distribution, emulsion and latex, and in various industries, and quality control of nanomaterials. So we will look this one after one. So for case of first one, water treatment. So water treatment and environmental science. DLS is extremely useful in water treatment facilities and environmental analysis. In water, we know there are many small particles like colloids, sediments and impurities. Even if the water looks very clear to our naked eyes, at the microscopic level there can be thousands of small particles which are harmful for the human health. DLS helps in measuring the size of colloidal particles, turbidity of water, stability of suspended solids, and the presence of pollutants in different size ranges. This is very important in wastewater treatment, river water monitoring, and in industries that need ultra pure water such as electronics, pharmaceuticals, and beverage companies. So this DLS overall helps ensure clean and safer water. And next is food industries. So for case of food and dairy industries, DLS has become a very valuable tool in the food industry, especially when dealing with emulsions and dispersions. Many foods are basically mixtures of tiny particles, like milk contain fat droplets, salad dressing have oil water emulsions, ice cream has proteins and air bubbles, cream and butter have different droplet sizes. DLS helps food scientists measure droplet sizes in emulsions, how stable the food product is, whether particles are aggregating or not, and how long the product will remain fresh. For example, if milk fat globules become too large, it means the milk is spoiling or separating. DLS can easily measure this. Similarly, in beverage industries, DLS helps ensure stability, smoothness, and consistent mixture. And further, it has a very major application in pharmaceutical industries also. This is one of the biggest application, as I earlier said. Like modern medicines increasingly use nanoparticles to deliver drugs. These include some of the names are like liposomes, micelles, nanoemulsions, protein formulations, and polymer nanoparticles. And one more term is mRNA delivery systems. For these systems, particle size is extremely important. This is why, because particle size affects how drug is absorbed, how long it will stay in the body, how it travels through blood, how stable the formulation is, whether the drug will work effectively or not. This DLS helps in measuring average particle size, aggregation of protein, stability of nanoparticle drugs, and its batch to batch tests, like are the medicines consistent or not. For example, with protein based drugs, even slight aggregation can cause harmful immune reactions. DLS helps in checking such aggregations in early stages. In short, DLS is a critical quality control tool in modern pharma. Next, we have chemical and polymer labs. Many chemical products are made of polymers, latex, and emulsions. Their properties depend strongly on particle size. DLS is used in chemical and polymer industry to study polymer chain size in solutions, latex particle size in paints, stability of polymer dispersions, emulsion droplet distribution. And in polymer research, DLS helps scientists understand molecular weight, degree of polymerization, how the polymer behave in different solvents, how their behavior changes, and this make DLS extremely valuable in R&D and quality control. Next application is nanomaterials and advanced materials. In industries that make nanomaterials like nanometals, silica nanoparticles, carbon nanoparticles, and quantum dots, controlling particle size is very crucial. This particle size helps to check optical properties, electrical conductivity, reactivity, color, and strength of the final material. DLS provides rapid, non-destructive measurements of these nanomaterials to ensure correct size, nano size distribution, and this is very important in paints, coatings, battery, cosmetics, sensors, and various emerging nanotechnologies. And further, moving to next applications, there are various applications or its relevance in our daily life, like cosmetic and personal care industries. DLS plays a major role in developing products such as face creams, lotions, sunscreens, shampoos, conditioner, serums. Most cosmetic creams are emulsions, and their smoothness, appearance, and performance depends on the particle size. DLS helps measure droplet size, stability, aggregation and life of the product. In sunscreens, nanoparticles of zinc oxide or titanium dioxide must be uniform and safe, and DLS ensures that. So cosmetics become safer, smoother and more effective because of DLS. Next is in paint ink industries. The color, gloss and smoothness of paint and inks are highly dependent on the size of pigment particles. Smaller and uniform particle gives brighter color, better coating properties, more uniform finish, longer shelf life, and DLS helps manufacturer maintain quality and consistency in car paints, printer inks, wall coating, industrial coating. If particle starts to clump, the coating becomes rough or the color becomes dull. DLS detects such changes in the early stages. And it is also used in quality control across various industries. In many industries, DLS is used as a routine quality control because it is very fast, easy, non-destructive and highly sensitive. It is very much sensitive to dust, particles and impurities. Various companies used it to check whether a product batch is consistent with the previous batches or not. If the particle size changes even slightly, the performance or stability of the product may be affected. And DLS allows it to check in early stages, and this helps us in decreasing wastage, improve efficiency and maintain standards. At last, in conclusion, I would say DLS is an accurate, very fast technique for nanoparticle size measurement. It is widely used in research, pharmaceuticals, chemical industries and water analysis. It is highly sensitive. It is very prone to dust and impurities, and its simplicity makes it a key tool to modern techniques. Thanks.